Alright, so now we're just going to go over question 5 and 6 in the packet. And so question 5 is just asking us to take a look at this picture and identify the different parts of it. And so we see here where these 4 circles kind of differentiate. Those are seminiferous tubules, and within them we have serotoli cells, and around them we have Leydig cells. And serotoli cells are what help produce the sperm cells. They kind of act like a nurse cell to help them grow up, basically. And so what we have first is we have spermatogonium, which is the beginning stages of a sperm cell, and then we have the spermatocyte, and then the older stages we have the spermatid, where it's ready to go through the right tubules into the epididymis and out through the vas deferens. And the Leydig cells are just in between these seminiferous tubules, and they kind of surround lymphatics and blood vessels and all that good stuff. And then so question 6, it asks us to list and describe the 4 thermoregulatory mechanisms utilized to keep the testes 5 to 6 degrees Celsius below body temperature. And it wants us to include the anatomical location of each mechanism. And so the first one is thermoregulatory neurons. These are located in the scrotal skin, and they regulate sweat glands, and they regulate the respiratory center. So they regulate when the animal is going to sweat and when the animal is going to start panting in order to cool down if they get too hot. The second thermoregulatory mechanism is the tunica dartos muscle. It contracts or relaxes in order to keep the temperature at the temperature it needs to be. And the third one is actually works with the tunica dartos muscle. The third one is a cremaster muscle. It also works to contract or relax the testicle in order to keep the temperature at 33 degrees Celsius. And then the fourth one is a pamepimform plexus, and that is where the countercurrent heat exchange occurs to keep that testicle at 33 degrees Celsius blood-wise. And so as the 39 degrees Celsius blood comes in through the artery, the 33 degrees Celsius blood is coming out through the venous blood, and as the venous blood is wrapped around that artery, it cools down that incoming blood and heats up the outcoming blood. And so it goes from 39 to 33, and then 33 to 39. And so that's how it keeps it thermoregulatory stable in the testes. And those are the 4.